we go. All right, we're up and live. Welcome back, everyone. This is episode, I think we're up to 13 now, Underscope Podcast. Today, I'm joined by Dr. Robert Mishur. What's going on, Dr. Mishur? How you doing?
I'm doing good.
I know you just finished up with some grading here, so we're looking to a well-deserved break for everyone. How you feeling about that?
I'm looking forward to it.
Well, the first thing I got to ask is, Dr. Mishur, your beard is crazy. It is absolutely ferocious right now. When did you start growing that out?
Probably right around the time I started at Widener, which was fall of 2016. I think it was just kind of stubble at that point. Yeah, been about 4 years.
I feel like that's really long for 4 years. I don't know. My beard would never get that long, but
well, you know what they say is if you're bald, you have a higher ability to be able to grow a beard. I don't know what the reason is, but I've heard that. So everything's kind of slid down.
Was there any reason you wanted to grow out your beard? You were just like, I'm gonna grow it out.
No.
You just wanted to? Do you ever think there'll be a day when you have to cut it off?
Probably. I mean, before I had this, believe it or not, I had a ponytail.
That'd be so
I cut that off right before I started at Widener, but you know, I'm bald, so it really doesn't look right. It's more of like a skullet.
Well, I hope that beard grows down to your knees. All right, Dr. Mishur, I definitely want to ask you, I want to start here. So you're from Chicago. What part of Chicago are you from?
I grew up in the South suburbs. And I was really young. I was in a city called Oak Forest. And then around 5th grade, we moved to an area called it was called Homer Township at the time, and I think it's now a city called Homer Glen. It's sandwiched between Lockport and Orland Park for those who are familiar with the Chicago area. And then I was there, pretty much until I went to undergrad.
What do you? I know you're a big Chicago Cubs fan, right?
Mm-hmm.
You always been a Cubs fan. Is there like certain districts that like make you a White Sox versus Cubs fan, or like just like whatever?
So I really wasn't into sports as a kid or growing up. It was really not until I was in grad school when I started to get into sports. In general, for Chicago, the South Side is White Sox and the North Side is Cubs.
Tradition.
But the suburbs are kind of free for all because I wasn't in Chicago proper.
Gotcha. Okay.
But when I was in grad school, I hung out with a bunch of people who were Cubs fans, and that became my team.
It must have been super relieving back in 2016 when they won the World Series. Was that the longest championship drought in sports history? I
believe so.
Okay,
at least in baseball, but I believe so.
That drought was incredible. I mean, it's crazy now. It's 4 years ago now that they won it. But what's one thing?
Like another one's on the horizon anytime soon.
Hopefully. I hope Chicago definitely needs some because the Blackhawks are always really good. The Bulls, they haven't been good for a while, but they need some love over there.
And then you had the Bears, who were doing great until we let Nick Foles start.
Well, hey, look, he won the Eagles the Super Bowl, so I don't know if you want to. I'm not, hey. What's something in Chicago that like a lot of people do that's overrated, and then something that not enough people do that's underrated?
That's a hard question.
Throwing you a curveball.
I mean, from a tourist standpoint, okay, I'm gonna go to the food example.
Go ahead.
The pizzas. All right. Deep dish Chicago style pizza is great, but people don't realize that the thin crust Midwest pizza is also special. And I haven't found anything like it out here.
Really?
It's the square cut, thin crust, but not too thin. It's usually dusted with cornmeal on the bottom. And everybody knows the deep dish Chicago pizzas, and then there's another one that not everybody knows is the Italian beef sandwiches, which is also a Chicago thing.
Really?
Shaved Italian beef, and they top it with mozzarella and giardiniera, and they dip it in the juice.
That sounds delicious. Holy crap.
I think a lot of people too, they go to Chicago for the first time. They do all the tourist stuff. They do downtown, Magnificent Mile,
Is the
Navy
bean
Pier.
there? Is that where the bean is? What's that bean thing?
The Bean is downtown Chicago. That's out by Grant Park, which is a big park out there.
What is that? Like, is there any symbol? Like, what is it even there for?
It's art.
Just art, just a big bean. Okay.
I mean, the Bean wasn't there when I was a kid, so
Okay.
it's art. It's a well-known landmark now, but it wasn't there when I was growing up.
Well, Chicago is definitely a city I definitely want to visit, for sure. It's super cold up there though, but I mean, I guess it's not that much different than
It
Philadelphia.
is, and that's one of the things that surprised me when I moved out to the Philly area, because we're roughly the same latitude as Chicago, but it's significantly warmer here, probably because of proximity to the ocean.
Yeah, I was gonna say that. Doesn't the lake somehow make it colder? I don't really know. Like,
Yeah, we have something in
sure.
Chicago called the lake effect, and it usually is a cooling effect.
Well, either way, I know I definitely want to at least visit Chicago once in my life. Definitely catch a Wrigley Field or something.
Wrigley's great. Wrigley Field, the area around there is all bars and restaurants. The Goose Island Brewery's there. Goose Island is originally a Chicago beer. I know there's a brewery in Philly now too.
Oh wow.
But yeah, and I hear they've expanded that area around Wrigley too. I haven't been back there in a while.
Okay, yeah. Well, maybe we'll go out there sometime. Once they allow fans back in, we'll catch a game. But yeah, so that's awesome. I definitely want to hit up Chicago sometime. All right, Dr. Mishur. So we'll hop right into this now. So I want to ask you a little bit of time about, like, I guess your undergrad. So I mean, you started as you were a chemistry major as your undergrad. Do you mind telling us like where you went and like how you wanted to become a chemistry major?
Well, to be honest, when I was a kid, I had some chemistry sets, and I was fond of burning things in the basement and mixing things together and seeing what happened. Back in the early days of the internet, there was something that went around, the text document called the Anarchist Cookbook, which had all sorts of interesting recipes in there. And I taught myself how to make gunpowder at home, which at the time you could buy all the ingredients at the pharmacy. I don't know if that's still true. Then I was fascinated by how you mix different chemicals in and you get different colored flames, and you put some copper in there and you get this blue-green flame. And so, I spent a lot of messing around in my parents' basement with that kind of stuff.
Hang on real quick. There's a skit, and do you watch It's Always Sunny in Philadelphia?
Absolutely.
There's a skit. I don't know if you remember this, but for those who aren't watching, there's a skit where Charlie Kelly mixes ammonia and bleach in the bathroom, and then he gets high off of it.
I don't know if you get high off that mixture, but
you would die.
Definitely get knocked out by it.
But well, I'm glad to hear that you were fascinated by mixing things together, and I think all chemists low key are oh my god, what's the word? Pyromaniacs? No.
Pyromaniacs.
Yeah, pyromaniacs. That low key, like not like any big deal, but so that was your fame to chemistry, just like mixing stuff together.
Pretty much.
So, okay, so you're in undergrad. Was there anything that stuck out to your time in undergrad? Like, well, hang on, where'd you go again?
I went to University of Illinois, Urbana-Champaign. So it's about 2 hours south of Chicago, out in like the farmlands.
Mm-hmm. And how was that experience, like overall?
It was a nice experience. That's where my dad went, so that's how I got drawn to the school. I had a lot of friends from high school that went there as well. Mm-hmm. It was a good experience. Ups and downs. I was not a good undergrad, I will say that.
Oh, what grade wise? Or you have to know
grade wise, behavior wise,
all
around. I was the student that I complain about now.
Well, hey, look, we all have that point in our life, and we'll get to that. We'll touch back to that a little bit to maybe advice to prospective students. But when did you realize that you wanted to go to graduate school and pursue like your PhD in chemistry? Well, specifically inorganic, but
Well, my first introduction to inorganic, it was one of the things that stood out to me for my undergraduate career. And it was an inorganic lecture. We were covering a topic called the spectrochemical series. And based on this, you could look at inorganic compounds and make predictions about their color. And I always thought that was so cool that you could just do a calculation on paper and then predict what color something is. And that's kind of what got me interested in inorganic. And I did a little bit of undergraduate research on molybdenum complexes.
Okay.
But then when I graduated, I didn't have good grades, and grad school wasn't even a thought in my mind. It was, I find a job. I didn't have a job lined up when I graduated. Went and spent a few months living with my parents in Minneapolis because they had moved north at one point when I was an undergrad. And then I ended up moving back down to the Chicago area, and I worked at a place called Cabot Microelectronics in Aurora.
Okay.
And what this place did was they made slurries, and these solutions, they look like milk, this white opaque solution. And what they're used for is polishing down silicon wafers to make microchips. And so I worked in a QA lab, and we get all these solutions coming in from production, and we do tests on them. And there were basically 4 tests I would do. I would do density. We had something called a densitometer, which is a black box with a tube sticking out of it.
Okay.
Put it in the solution, press a button, it pumps some through, and it spits out a number. I did viscosity, which is, you know, the spindle that spins, and you just put the cup up underneath it, and then you write down the number it spits out. Percent solids, so weigh it and evaporate it, calculate percent solids. And I feel like there was one more test in there, but
let me guess, you got bored of doing the same thing every day? You got bored of, like...
Very much so. It was very repetitive. I was pretty quick at it. I was working second shift, so like 2 to 11 kind of hours.
Okay.
And, you know, I got caught, when I came in, there's usually a backlog from the end of the previous shift. But once I caught up with that, I just had all this idle time, and I was sitting around playing Defend Your Castle, the old Flash game, wasting time because there was nothing to do.
Yeah.
So bored with it. And one of my friends was in grad school, and he said, "Hey, why don't you come to grad school?" I said, "I don't have the grades for that." I mean, yeah, you can still get in here. And so I went, and after a year off, I went and did grad school and managed to fix my behaviors and get better grades and kind of get things together.
Well, hey, look, a year out from, I think, of university definitely like puts some perspective on it. And I guess like taking your time, like in a real job, doing the same thing every day, you just had this like epiphany. You're like, screw this, I want to go do graduate school.
Yeah, I wasn't a fixed hours kind of person. It wasn't right for me, and yeah, like I said, I was bored with it.
So now you're at graduate school. This is where I think this is probably the most fascinating about you. So you got your Ph.D. in inorganic chemistry. Okay.
Right.
You're always fond of that. But you did your postdoctoral work in organic chemistry and some, I assume this is biochemistry as well, your mechanism.
Biochemistry / biology. Yeah.
Right. So, well, first of all, I want to ask about your what was your thesis and your dissertation about for inorganic chemistry? Like what
I didn't write a thesis. So I never got a master's degree along the way. I went right from bachelor's to PhD. So my dissertation, I worked with platinum compounds. So it'd be easiest if I can draw this. Can I share my screen? And
Absolutely. Okay. Yeah, go ahead. Let me see if I can give you privileges real quick.
Yeah, you need to give me privileges.
Get it here. Let's see. Okay, I'm just gonna make you the host for now. There you go.
Okay. So there's a compound. Have you heard of a compound called Cisplatin?
Probably. I don't remember off the top of my head, but
okay. So here's the structure of Cisplatin. It's a square planar compound with 2 chlorines and 2 ammonias on a platinum.
Okay.
And this is a rigid molecule. So the 90-degree angles, no real flexibility there. Right?
Okay.
In water, these chlorines pop off. And then platinum seeks out bases, and it likes nitrogen. And specifically, it'll bind to the N7 positions of guanine and adenine, two of the DNA bases. And what happens, I'm going to try to draw a double helix, and I know this is going to not look like a double helix. Oh, okay. So if you have two nitrogens on this strand, the platinum will attach there. Okay, these two chlorines will come off and it'll attach to those two nitrogens. But since you have this rigid geometry around the platinum, it'll curve the DNA around it so it can do this. And so the DNA gets curved around the platinum and it forms basically a kink in the DNA strand. And this interferes with processes like transcription and replication, and it leads to cell death. So this is an anti-cancer drug. It's specifically, I mean, it's toxic to all cells, but cells that are rapidly dividing are the ones that get, hit. This has been around, I think it was discovered in the 70s. A big drug for treating testicular cancer. Testicular cancer was nearly always fatal prior to the discovery of this drug. And now it has one of the highest cure rates. It was the drug Lance Armstrong was on as well.
Oh, wow.
And up to this point, people didn't really think there was medicinal value of transition metals. All the drugs were organic chemicals. But there's a lot of problems with cisplatin, specifically with side effects and dose-limiting toxicity. So people have made a lot of derivatives of this compound to try to reduce the side effects and the toxicity.
Okay.
And so I was making derivatives that had a pyrophosphate group on them. So you'd have
this is really cool.
Something that looks like this on there.
I'm sorry, you said you were trying to synthesize these, right? This is what you were trying to synthesize?
Yes, we were synthesizing platinum groups that had this pyrophosphate backbone on them. And then here you'd have some nitrogen groups. So it could be an ammonia. It could be some other things, like we used ethylene diamine.
For those viewers right now that are just listening right now, basically you have this, Dr. Mishur just drew like an oxygen phosphorus ring around the platinum, connected to the platinum. That's essentially what it is.
Yeah, and so this is an ethyl group on the left. So that's ethylene diamine. We also did diaminocyclohexane. It looks kind of like that. Obviously, I'm not drawing this to scale because I'm drawing on a vertical board.
So in water, which side pops up?
It's the pyrophosphates that's the leaving group.
Oh, okay.
So those nitrogens are kind of stuck there.
Gotcha. Okay.
So,
and the other side, so those nitrogen groups, those are the ones that cause the kink in the.
This part of the structure doesn't change. The part on the right comes off, and that's where the nitrogens and the DNA attach.
Gotcha. Okay. So whatever you're putting on the right side of this molecule is essentially what you want to be non-toxic, right? That's what you want to be.
The non-nitrogen parts. Well, yeah, you want that to be non-toxic, but you want the whole thing to be non-toxic.
Well, yeah.
Reduce toxicity.
Okay.
So
what was the issue with the Cis Platinum? It was because the chlorines were like such an issue?
It's because it isn't specific for cancer cells. It can target any cell. The dose-limiting side effect for cisplatin, it's been a while. I think it was liver toxicity.
Okay.
But you also get all the other fun chemo side effects like hair loss and tinnitus and various other things.
Okay, so in theory, I guess the theory is that depending on what nitrogen groups you put on and whatever you have on the other side would dictate what cells it attacks?
Well, it seeks out DNA, and there's evidence that it interacts with proteins as well.
Okay. So were you specifically, so you were just trying to synthesize this, right? You weren't necessarily interested in getting it to attack to certain cells, right?
Well, I was doing synthesis. We also did kinetics experiments to see in water, how long does it take for that group to come off, that pyrophosphate group? We were doing a lot of, my boss at the time was a big NMR person, so we did a bunch of inorganic NMR. We did phosphorus and platinum NMR to characterize these and to do the kinetics to show the rate at which that group comes off.
Okay.
Someone else in the lab did some cell work, and we showed the toxicity of this, and I believe it actually had improved toxicity over cisplatin in some cell lines.
Okay.
One of the issues with cisplatin, besides the toxicity and side effects, is resistance. And sometimes cells can acquire resistance to cisplatin, sometimes it's inherent. So we took a cisplatin resistant cell line and we showed that these compounds were more effective in a resistant cell line than the parent drug.
That's awesome.
I wasn't doing the cell studies.
Okay. So what did you like? So at the end of your at your dissertation, like, what was the conclusion? Like, were there a couple of conclusions that you were able to make?
Yeah, I mean, we made several of these compounds. There's a number of compounds that I made. They showed improved action over cisplatin in certain cell lines. My boss ended up founding a company to further investigate these, and several of these were patented as well.
Wow, that is awesome. I see when people talk about their work, it gets me excited for graduate school. So this is really cool. So I guess obviously, so the next question is like, so this your work in your dissertation, your work in graduate school got you prepared for your first postdoc, then, right?
Right.
Was there any reason why you wanted to go pursue a postdoc before like going back into work, or is it just like you just wanted to keep doing research?
At the time I was set on, I'm gonna go and be a professor and run my own lab, and that's what I wanted to do. And so that's the route, because if you're obviously that's not where I'm at. So some things happened along the way that we'll talk about, but you know, that's the next step. You do your PhD, you do a postdoc, which places that offer tenured positions, they generally look for you to have that.
Right. Are postdocs being phased out? Like, I mean, I guess if you wanted to go be a professor, you need postdocs, right? Like,
I'm not under the impression they're being phased out.
Okay. Okay. Well, because I have interest in teaching, but I guess
now, tenure, on the other hand, is slowly getting phased out. So
how is that being phased out? Like getting tenured? What does that even mean?
Well, tenure basically means you're employed and the university can't release you without a really good reason. It's basically job security.
Okay.
Okay.
How is that being phased out? It seems like a good thing.
Well, by hiring people that have positions like I do. I'm in a non-tenure track full-time position. And by over-reliance on adjunct instructors.
Oh, okay. That makes sense, because I know adjuncts don't get paid a lot, so they do a lot of work. Okay, but so going back to now to your postdoc though. Okay, so you did your work, and it kind of aligns a lot with like organic and like pharmaceuticals. So you took your first postdoc was in supramolecular organic chemistry. So what specifically did you do, and like what does that even mean?
Well, let me tell you how I got there. So I really wanted to keep doing the platinum work. I was really into that. And I had a really hard time finding someone to take me, because it's in that bioinorganic realm, and I had the inorganic background, but I didn't have the bio background. And despite that, I wanted to do the cell work. My boss put it off on someone else, and I never got that experience. So I had a hard time finding a group to take me. I had applied to a group, or I'd written a letter to a group at Tulane in New Orleans. There was a group doing platinum work. It wasn't the cancer stuff, but it was still platinum chemistry. And he wrote back and said, I don't have a job in my lab at this time, but there's this other guy I work with who's looking for someone. Let me send you his name. And this other guy, calls me out of the blue and says, I'm a professor at Tulane and I'm looking for someone to come work in my lab. And we talked about it. Absolutely nothing to do with my PhD.
Okay.
So I went down to Tulane and I spent 2 years there. I can show you what I was working on.
Yes, please.
So we were working with these molecules called cyclodextrins. All right. Now, all you need to know about cyclodextrins is it's kind of got a truncated cone shape. It's hollow in the middle, and you can encapsulate things inside. It's a cyclic sugar molecule. Okay. And so this is like this truncated cone with a hollow interior. And because of that, you can stick things inside of them. And so you could do things like take a drug and sandwich it between two cyclodextrins. Okay. And incidentally, this is the active component in Febreze, these cyclodextrins, because they basically pick up order molecules and they get trapped inside these things. So the idea here was we're going to take a DNA hairpin loop. So you have this hairpin loop. Excuse my art. These are all
you didn't go to art school, so.
Yeah, these are all base pairs up the neck, okay? And then this is just a single strand of DNA, all right? And what we wanted to do is on the ends of this, attach these cyclodextrin groups. The idea is we could trap a drug between these, and then if we bring in the complementary strand to this DNA, the hairpin would unravel and then the drug would be released. And we know that in certain cancers, you have these small RNA chains. And so if we make this complementary to one of those small RNA chains, you could theoretically target a cancer cell. And where that RNA is being produced, this would open up and it would release the drug. Okay, that was the goal of the project. Now, we didn't really get there. It was
like many other things,
a 2-year experiment in futility. The main issue we had is we needed to be able to see that the drug was going in and out. We needed to be able to prove we brought in this complementary strand and it released the drug. Okay. We chose a drug called CD437. It had a planar aromatic ring on one side and something called an adamantyl group on the other side. Okay. And we showed, so the first step is to show it can do this kind of 2-to-1 binding or stick in the inside. All right.
How did you prove that? Like NMR and like X-ray diffraction? NMR, okay.
NMR. Then the question is, can we open this up and show that the drug's being released? And so NMR, we also used fluorescence, okay, to do this. Now, the problem is you need some sort of spectroscopic handle. And to do this, we had this large, flat, planar aromatic region on this molecule, which would fluoresce. So it's great. We can monitor it. There's a change when it's encapsulated versus whether it's out. Okay. We could, again, show that it has this 2-to-1 binding. But when we open it, we didn't get the results we expected. Okay. We'd expect that, it'd be you introduce the DNA, it opens up, and then you get this change in the fluorescence. The problem is because you have that flat region can slide between two DNA bases and percolate into the DNA change. And then it's still shielded from the surrounding water. So because we had that group there and we needed that to measure this with fluorescence, it was also being a problem because it was slipping into the DNA helix. So we never was able to conclusively show this, unfortunately.
Out of curiosity, are they still working on this now? Or
I don't think so, but it's been 10 years.
Yeah. Okay. Well, I think this is still pretty cool.
The super molecular stuff, it's about self-assembly, large-scale self-assembly, and it's a lot of really cool organic stuff there that you don't really get exposed to at the undergraduate level.
So what was the purpose though of like, because it just seems like it almost kind of reminds me of like a pill. Like if you have these two, if you have the 2-to-1 binding, you have two halves and you just put something inside of it. Why not just deal with the pill? Like what?
Well, the idea is it'd be targeted because the RNA that you would need to unlock this is something that's produced by certain types of cancer.
Okay.
So we want to have targeted release in the cancer area.
Okay, so let me try and get this straight. So in theory, cancer releases this RNA that would unlock the drug that you have.
It would cause the hairpin to unravel. Yep.
Oh, and then what? So then in theory, whatever was inside of the I guess what do they call it? What were they called again?
Cyclodextrin.
So whatever was inside the cyclodextrin would be unlocked by the RNA, and then that would attack the cancer?
That's the idea, yeah.
Oh, okay. That's really cool. Shame it didn't work, but
yeah,
or at least didn't really get inconclusive, but people are still working on it. Do you think that there is a solution to cancer yet, and no one's really, it's just not monetized? Or
I don't think the solution to cancer is strictly in chemistry.
okay.
There's not going to be a one drug cure all that's based on a specific chemical molecule.
Mm-hmm.
The future of cancer therapy is the CAR T therapy and the individualized cancer therapies, right, which are immunology based.
Yeah, I have a lot of friends that ask me because, like, you know, I bring on like Elena Fadakova. Mm-hmm. They do. I mean, obviously in chemical engineering, they do cancer immunotherapy stuff. Like, there's so much cancer research, and people ask me, like, you know, like, is there a cure to cancer? I'm like, well, first of all, it's not like one size fits all. Like, every cancer is way individualized that you wouldn't even believe. Even if there was, the question is, you know, is there money to be made? You know, people charging people to cure cancer. So I always say, like, I don't know. That's a tough question, but I doubt it. I don't think there's a solution yet to any cancer, but we'll see. Okay, so then then you did your second postdoc. So how did you arrive to your second postdoc, and then what did you do in that one?
Well, first postdoc was done, needed a job. I applied to a variety of things. I applied to industry positions, I applied to postdocs, I applied to faculty positions, and this one just popped up, and it was in Texas, in San Antonio. And this guy's studying aging, and he's doing mass spec stuff, and he has somebody that's working for him, and he's about to leave. I need somebody that can do some mass spec. So he's looking to hire a chemist, even though he's running a biology lab. And I didn't have any other offers. So packed up, drove 8 hours down the road, and ended up in San Antonio.
Hang on, real quick, before you go into this, San Antonio, I hear, is a beautiful city. Like, I hear it's an awesome city. Is that true?
San Antonio's a nice city, yes.
Nice? You wouldn't say it's breathtaking?
Parts, sure.
Sure. Okay, fair enough.
San Antonio's an interesting mix. So there's a lot of military there. I think there's 3 bases. There's a couple Air Force bases and an Army base in San Antonio. You have a big medical center in North San Antonio, the UTSA, and then I was actually part of the UTSA network. No, I was part of the UT Health Science Center network. So you have UTSA and the UT Health Science Center. And so up in like North San Antonio, you have a bunch of, you know, college people and medical professionals, and it's largely Hispanic, and there's a significant amount of German population in the area surrounding San Antonio. So it's an interesting kind of blend of things.
Is that where the Longhorns are, the Texas Longhorns?
No, that is not. That's Austin. About an hour and a half north of San Antonio.
Gotcha. Okay, never mind. Okay, so then you're in San Antonio doing, well, biology, biochemistry work. So
Yeah. So yeah, I went from New Orleans to San Antonio. Big difference from New Orleans. New Orleans is just crazy.
is that good or bad? I guess it
Not
depends.
so good for productivity.
Depends on who you ask.
So yeah, I end up in this lab, and this guy's studying C. elegans.
Okay.
So
What are those?
C. elegans, it's a nematode. It's basically a little tiny worm. The adults are about 1 millimeter long. You can barely see them with the naked eye. You can't see the eggs and the larvae with the naked eye. It's a really widely studied organism. I know you guys are all familiar with Drosophila.
The fruit flies. Woo!
Yeah, C. elegans is, I would say, as widely used as Drosophila.
Okay.
Okay, it's a very common organism. And it's easy to do genetic manipulation in them. So I did a couple of things. First of all, I was just the all-around, this is too expensive to buy, make it for me guy. I actually have my name on a paper where I did nothing other than synthesize one of the chemicals for a research group, because they knew I could do the synthetic chemistry. But the main brunt of the project was doing something called metabolomics. And so the idea here is you can culture these worms in a buffer solution, like phosphate buffered saline. And they excrete metabolites, basically the end products of the different metabolic processes. And they spit these out like directly into solution. So then we can suck up the solution and shoot it through GCMS and look at what chemicals are in there, okay? And what we were trying to address was there is a variety of genetic mutants that have disruptions somewhere in the mitochondrial electron transport chain. And several of these are long-lived compared to the wild-type animals. The wild-type animals live, it's been a while, I want to say around 21 days. And you can get significant life extension with some of these mitochondrial mutants, which begs the question, well, mitochondria is good. Why is screwing with the mitochondria leading to lifespan extension? Okay? And not all mitochondrial mutants have lifespan extension. It's only certain ones. Okay. And so we're analyzing the chemicals these worms are producing and trying to trace backwards the metabolic pathways and figure out what pathways were disrupted and why. And the proposal was that, is it possible that this pattern of chemicals, well, let me back up a sec. Sure. We found that several of these long-lived mitochondrial mutants produce the same pattern of chemicals.
Okay.
And this is different from non-long-lived mitochondrial mutants, and it's different from long-lived mutants that don't have a mitochondrial mutation.
Okay.
So there's something that's unique about this set of long-lived mitochondrial mutants. They're all producing the same pattern of chemicals. And so we were postulating that maybe it's the chemicals that are responsible for the lifespan extension. We were able to figure out why they all produce the same pattern of chemicals, and now we were trying to figure out why they lived longer. And so then we were going and feeding mixtures of these chemicals back to wild-type animals and trying to see if we could extend their lifespan just by feeding them this chemical cocktail. So results were
varied, probably.
Yeah, varied. We achieved modest lifespan extension with some of the chemicals and some of the combinations of chemicals. So that's kind of where that was left at.
Well, is there any further? Have you touch base with them since you left it, or
they're no longer doing metabolomics. So
it's probably too complicated. It's probably too complex. How do you how do you keep one thing controlled and study those things? You know,
it's very complex. One of the issues is the way you culture these animals. They live on an agar plate, and there's this film of E. coli on top. and they just crawl around the surface and eat the bacteria. That's the food source for them.
Mm.
So the question now is, how do we drug them? Do we add these chemicals directly to the plates? And if so, how do we know that they're being metabolized by the worms and not by the bacteria on the plate?
Mm.
So yes, it is very complicated. Lot of variables. I mean, temperature is a variable. When you keep all the worms in a temperature controlled 20-degree fridge, and yeah, it's the biological organism. It's very complex.
Well, what I think is honestly most fascinating about what we've been talking about so far is first of all, your journey from a a very nonchalant kind of a screw off student, to now like 2 postdocs, a PhD. What do you make of like that journey? Like, what do you like? You look back and it's like, wow, look at all that I've like accomplished. Like, what do you think? What do you make of it?
I wish I'd done more.
Yeah. What do you mean by more? Like in your undergrad, your graduate, like just
In my undergrad. I wish I had not screwed around as much, gotten better grades. It would have opened more opportunities for graduate school.
Mm-hmm. Wow. So then the next question is, how did you end up at Widener, right? So you finished up your second postdoc, and then I guess you're here all of a sudden? You just popped in?
Well, so the second postdoc, my boss didn't have tenure yet. So he was still, a young faculty member trying to get tenure. Mm. And I would come into the lab in the mornings, and he would be asleep in a stool, curled up somewhere in the lab, because he was sleeping at work. Wow. He was still at the bench doing experiments himself, in addition to writing grants. And I said, I don't want to do this. And actually, one of the professors at Tulane told me that when he was doing his postdoc, he was working over 100 hours a week. And I said, I don't want to do that. So, what the heck? I kind of lost wanting to go to an R1 institution, a research heavy institution, and do that kind of work, because it wasn't the work-life balance I wanted.
Sure.
And so, plus on top of that, I was not a fan of writing grants, and I didn't have any grants under my belt, which really hurt my competitiveness trying to go for a tenure track job at an R1 institution. The other issue is my diverse background is more of a liability than an asset. Really? Because they want people who are experts in one subject. I mean, that's kind of what you're looking for a tenure track position. You're supposed to be an expert in your field, and they're looking for people that, are an expert in one area versus I know a little about this, I know a little about this, I know a little about this.
Sure.
And so I remember when I was in grad school, I was a TA for the entire time I was there. And I actually taught inorganic lab and PChem lab.
PChem.
Yeah, I was a PChem lab instructor. And I was really good at it. The undergrads liked me. At least I thought I was good at it. And I always liked that. So I started to look for opportunities in teaching. And at the time, I was chairing the San Antonio section of the American Chemical Society. I had some ins there. I got a few adjunct positions at some different schools in San Antonio. And once my postdoc ended, I was teaching 7 classes a semester, which sounds insane now, at 3 different schools. And I was driving between the 3 schools in the San Antonio area.
Holy Christ.
Yeah, it was pretty rough. But then my girlfriend at the time, my wife now,
nice,
finished her PhD, because she was getting her PhD while I was doing my second postdoc at the same school. So that's how we met. And she got a postdoc offer at Children's Hospital of Philadelphia.
Wow.
And so we said, well, I'm adjunct teaching. I can do that anywhere. So might as well move out to Philadelphia. And so we moved up to Manayunk in 2014. I got a 3/4 time teaching position at Richard Stockton, which is in New Jersey, almost all the way out to the coast.
Yeah, I was gonna say that's like isn't it like an hour and a half ride?
It's about an hour and a half commute from Manayunk.
Oh my God.
Fortunately, I became friends with one of the people that was teaching there, and he was driving from Ardmore, so we had the same commute. So we were able to split that.
Okay.
So I taught there for a year. Then once that was over, I went and I worked at Manor College in Jenkintown for a year.
That's like an hour in the other direction, isn't it?
No, Jenkintown was about 30 minutes from me.
Okay.
But then after a year, they let me go, and they said it was due to a drop in enrollment. No, I'm not going to theorize on that.
We'll let that one be.
It's a very small school. They
Sure.
had 2 chemistry faculty. They decided they only needed 1. And we'll leave it at that.
Well, we're off to better places anyway.
I am glad I am no longer there. Let's put it that way. And I found the job at Widener. That was 2016. I found the job at Widener, and I've been there since. And I really like it at Widener, so I'm happy to be there.
That's awesome. Well, it sounds like a crazy ride for you. I remember. Oh my God, there was before COVID. There was it an ACS thing that your wife had brought into Widener, and we were all there. I forget what it was.
Oh, it was.
I forget what it was.
My wife and I, at one of the president's welcome back parties, we ran into Dean Lehman, the arts and sciences dean, and he had invited her to come and give a talk on her background and her career path, and it was kind of an unofficial part of the science seminar series that had its own special time and date. But yeah, she had a chance to come and talk at Widener. She is a scientific editor for American Association of Cancer Research, a journal called Cancer Immunology Research. And so she does work with them. That's
incredible. What was her PhD in? Was it in chemistry?
My PhD was in inorganic chemistry.
Her PhD.
Oh, her PhD is in microbiology and immunology. She's an immunologist.
And then okay. That's cool. I was just curious. So now you're here at Widener. What are some like your favorite moments here? Even though you only spent 4 years here, so.
Yeah, I've only been here 4 years. I mean, I taught 390 for the first time last year, and I really enjoyed that. I'm really looking forward to teaching 375 next semester because I have never taught inorganic. So
just crazy because your PhD's in that. So
it is crazy. And part of the reason I got my PhD in inorganic is I really hated organic when I was an undergrad. Like, organic 2 was not my friend, and my naive mind thought that inorganic was the opposite of organic, which is not true at all. So I said, I'm going to be an inorganic major. And ironically, I've taught organic labs and lectures numerous times at numerous places and never inorganic.
That is funny. Oh my God. I thought inorganic was the opposite of organic. That one's going down. I'll have to keep that one in the record book.
No, I've really enjoyed it at Widener. I trying to think of anything specific, but I have enjoyed my time there.
Yeah, just overall. What do you think? Let me ask you this, because I mean, I had you for Gen Chem 2. It was just, oh man, when even was that? 3 years ago, 2 years ago?
Something like that, yeah.
What do you think some of like the misconceptions are like about you, and like you want that some of your students you think that should know about you?
Well, I always get in my course evaluations that my students think that I'm inapproachable. And I don't feel that way about myself, so I wish my students wouldn't feel that way. I do occasionally have a low tolerance, and I certainly have high expectations, but it comes from the fact that I don't want my students to repeat my mistakes.
Right.
And I see a student acting the way I did when I was an undergrad, it bothers me because I don't want to see somebody else go down that path. And I try to, talk to students that are struggling and phrase it that way, that, I know where you're coming from, but you really need to make the life changes now, because, it comes to a point that it follows you. I didn't get into a top tier graduate program because I did not have top tier undergraduate grades. Right. I did not get the best postdoc because I did not go to a top tier graduate program. And it follows you.
Right. Did you teach Gen Chem 1, or was it just Gen Chem 2? Was it both?
I teach both.
Okay. Yeah, I mean, it's tough because I think first of all, I think a lot of chem faculty get that. Like they seem like they're inapproachable. I don't know. Like I mean, like I was never afraid to ask questions. Like I'm not afraid to like do anything. But so I'm different in that way. But I guess for the common student, like it's tough because first of all, I think Gen Chem in by and large is already like a tough subject for a lot of students, and I wish it weren't, but it is. So there's that. So people are already feeling down about that, and then like they get nervous to ask the professors questions and stuff. But what do you think? Like, what do you think that we can do as like to like alleviate some of the pressure that goes on with like Gen Chem? Because a lot of students get really like bogged down by it, but I don't think it's that difficult.
Well, it's a mixture of things. I mean, what I would say to the freshmen is go to office hours.
Right.
We have the chemistry tutor center, and then we all have office hours, and they are so underutilized. And that really is the best thing for struggling students to do.
Is there any advice that you would give to like prospective students and students in general, if you could circle back to everything that you've learned?
Give it your all. You only get one shot at it. I mean, that's my advice. You only get one shot at it. So
make the most of it for sure. I try to live that every day.
And in hindsight, I didn't think that way when I was younger, and that's really, because it's still in high school mentality. And for me, my big problem was I got through high school, top 10% of my class out of 700 plus students, took a bunch of AP classes, math through calculus, I took chem, bio, physics, and I didn't open a book in 4 years.
Right.
And I didn't have study habits because I never needed to study. And then I get to college and, I don't have any study habits. I don't have any study skills, and I can't skate by anymore. And everything comes crashing down.
Isn't that so fascinating? That's always been so fascinating to me that you can skate by, like, from even for myself, like I skated by like AP chemistry. Now AP chemistry was different. It was easily the hardest class I think in high school, but I passed it with an A without even opening a book. Like, so it's crazy to me how that design, and then you get hit. I mean, you get smacked right in the face, I think, first, like freshman year. And in a lot of ways, you cut the baggage. Like first and second semester of chemistry will cut the baggage. Like it will cut people out if you're not ready for it. And I think it's truly a disservice to chemistry everywhere because I don't know. Like I think everyone should have at least a basic knowledge of it. But I don't know. It gets shafted as like this like terrible subject. Crazy to me. Let me ask you this: what was like? So you've traveled a lot around the United States, like being in like your undergrad, post grad
Hmm.
docs, and now you're here in Manningham, Philadelphia area.
Now, actually, I'm in Delaware now.
Okay, you're in Delaware.
We're in
Where are you in
Delaware now.
Delaware? Wilmington.
We're in Claymont. We're literally just over the state line off I-95.
Okay. Are you by the big fine wine and spirits or the?
I can walk there. I'm right behind it.
I love that store. That store is great. What do you think? What's like been your favorite city that you've been to? I'm also going to include. I don't know if you travel anywhere, but I also want to include those cities too.
I've traveled a lot. New Orleans was a blast. I don't think I would ever want to long-term live in New Orleans.
Mm.
It is certainly not a place I would want to settle down. But
Is Mardi Gras overrated, or is it actually really fun down there?
it's a blast. So let me tell you about New Orleans. And it's a blast. And I was there right out of grad school. So I was, when I moved there, 20, I was 27 when I moved to New Orleans. And
prime time for New Orleans.
It was great. Old enough to drink and a little bit more mature.
Yeah.
And so I get down there. I meet my neighbor. I'm in this like fourplex apartment complex. Neighbor goes, "So you gonna go to Muses tonight?" What's that?
Yeah, right.
Oh, that's the all woman crew. That's when they throw the shoes. They're even rarer than the coconuts. And I'm just looking at them like,
wait, what?
And a word of this. So sure enough.
So now you gotta go, I guess, right?
The parades for Mardi Gras start about a month before actual Mardi Gras.
Oh my god.
And they run like once a week, and then they start picking up, and then they'll start running 2 or 3 a night. The Thursday before Fat Tuesday, they have an all-woman parade crew called Muses. And everybody hand decorates a pair of high heel shoes with glitters and sequins and stuff. And they toss them from the floats like they do the beads.
Okay.
And people fight over them. My friend dislocated his shoulder trying to get a shoe. Because they're collector's items.
Oh my god.
And then on Fat Tuesday, the parades start at 8 in the morning. The first parade is Zulu, which is the African American parade. Okay. And they hand decorate and throw coconuts from the floats. And people fight over them.
Did anyone ever explain to you why they do that? Is it like a cultural thing, or are they just random, like super random? They just
I don't know all the history of all the different Mardi Gras crews. There's a bunch of different crews, and they have their scheduled times and their parades. And I lived a block and a half off the parade route in Uptown. So not in like the French Quarter. I was in Uptown, New Orleans. Mm-hmm. And for Mardi Gras, everything kind of shuts down. Like the Fat Tuesday, the entire town is shut down. Schools are shut down. The post office is shut down.
Did you take off? I assume you took off grad work those days.
You can't get anywhere because of the parades and the massive amounts of people. In the middle of St. Charles, which is the street I lived off of, they call it the neutral zone, but it's big grassy median. Mm-hmm. People would set up tents, put kegs out, and just camp out there and hang out. And it was just nuts, like flat out pandemonium.
Wow. And that sounds really fun.
Yeah, it's fun. St. Patrick's Day, they throw cabbage and carrots from the floats.
Are those designer items too?
I don't think so. I think that's just cabbage and carrots. It's just such a wacky town. Yeah. And I didn't live near the French Quarter. I had to, a few miles away from that madness. But yeah, still, it's pretty crazy.
Out of curiosity, you ever been to like a Saints game? Did you ever go to one of those?
I've been to the Superdome, but not for a Saints game.
Okay.
So we saw Tulane used to play their games at the Superdome.
Okay, because I know the Superdome is like, that is like, it's like incredible.
And when I was there, that was when the Saints won the Super Bowl. So
holy,
Saints won the Super Bowl. They had the Saints Parade, which was also during Mardi Gras season, so we shoved another parade in there.
Oh my
We
God.
actually went to that. We went to the Saints Championship Parade.
Dude, that sounds like it sounds like the Philadelphia parade, but like on steroids. Like, oh my God.
New Orleans does partying to the max. That is for sure.
Did anyone tell you why they celebrate Mardi Gras? Like, why? Like, is there any reason why they picked Mardi Gras to be like there? Like,
I don't know. I do not know. I mean, the city has Spanish and French influences. It's got an interesting history.
Mhm.
Surprisingly, St. Louis also celebrates Mardi Gras. I believe they have the second largest one in the country.
Really? Okay.
Yeah.
Have you like traveled anywhere else, like vacation? Like, where else have you traveled to?
I've been over most of the country. I think I'm missing 7 states. So
Are 2 of them Alaska and Hawaii?
What's that?
Are 2 of them Alaska and Hawaii?
I was in Hawaii when I was 8, and we were supposed to go to Hawaii in this last May, and that didn't happen.
Yeah, well.
I was also supposed to go to Italy and Germany this past summer, and that didn't happen. So
I'm sorry to hear that one.
Yeah, COVID really messed with travel plans.
Did you at least get a voucher? When this is over, can you?
Well, American Airlines gave me a 1-year voucher, and I'm going to call back and go, it's been a year and I still don't want to get on an airplane. What can you do? And we'll see. We'll see what they say. But yeah, New Orleans was definitely a fun city. Other places I've been, I mean, I've traveled a lot for the ACS conferences.
Yeah, is there anything like stick out? Like, what's like a,
San Francisco was a lot of fun. I know my second year anniversary with my wife, we went to Breckenridge, went skiing in Colorado.
You're a skier?
No.
Oh, are you a watcher as your wife skis?
I skied a few times in high school. Some of my friends and I drove out to Vermont and went skiing in Mount Killington or something. I had never been in skis before, and my idiot friends put me on a black diamond with moguls. And
no, it was black diamond.
Didn't know what a black diamond. Didn't know my ratings, and I certainly didn't know what a mogul was, and I certainly didn't know you're supposed to go around them and not over them.
Oh my God! Oh Jesus!
So,
wow. Yeah. Well, Dr. Mishur, I want to thank you so much for hopping on the show. First of all, you got a lot of stories bottled up in there. I definitely want to have you on here again sometime in the future. We can expand on those stories, and all your wisdom, all your experiences. I want to thank you again for coming onto the show.
Oh, absolutely. Thank you.
So without further ado, I mean, if you like the content, please make sure you like and subscribe. Dr. Mishur, he'll be here, Inorganic, next spring. So get ready for it. Alrighty, everyone. Thank you. Unless I start